Good evening. Welcome to UT Brainstorms. My name is Mike Mock. I'm a professor in the Department of Neuroscience at UT Austin. Welcome to Brainstorms number 40. This is the fourth Brainstorms of our fifth season. So as always, we want to thank you for your interest and continued support. We enjoy doing Brainstorms and we hope you will continue to appreciate them. As always, I want to begin also by thanking Laura and Elena, who make everything happen here. Thank you so much for everything, and we'll have a special thanks for Laura a little later. So we enjoy doing Brainstorms, and we sponsor it here in the Department of Neuroscience, in large part because we enjoy sharing our passion and expertise about neuroscience with you, and we learn and enjoy hearing from you. We learn from your questions and such. And so a very important part of Brainstorms is the question and answer. And so the format, as is always the case tonight, will be, we'll hear from our speaker, Franco Pistilli. And then when he's finished, we'll have a panel of UT experts who will be here to answer your questions. You can type in your questions into the YouTube live chat and feel free to begin that even as Franco is speaking, and then we'll address as many questions as we can up until quitting time. So it's a pleasure tonight to introduce Franco Pistilli, who's an associate professor in the Department of Psychology and a member of the Neuroscience Graduate Program at UT Austin. Franco's relatively new at UT. He's been here, I think we recruited him maybe a year and a half or so ago. It was during COVID, so it couldn't have been that long ago. And again, as I've said with a couple of our previous speakers, the fact that we're able to recruit people of Franco's caliber is just an indication that the already strong Department of Neuroscience just keeps getting stronger. So we're very happy to hear from him tonight and many members of his lab as panelists. And he's going to be talking to us about sort of a modern approach to neuroscience. And so, without any further delay, let's do that thing that we do when we can't be there in person to clap, and somehow find a way to welcome tonight's speaker, Dr. Franco Pistilli.
Hi, Mike, and hi, everyone. And thank you for the kind introduction. I'm honored to be here.
Unmute yourself.
I am unmuted. Do you guys hear me? No?
We still can't hear you.
How about now?
Not yet.
How about now?
Not yet.
I think other people can hear me, and I can hear myself.
Mike,
maybe it's a problem with your audio?
It was me. Apologies. You go.
Good. Well, let's start again. Thank you for the kind invitation, and I'm honored to be here, both at UT and speaking and joining this event with you guys tonight. I am associate professor in the psychology department here at UT, and I'm also affiliated with the neuroscience department and the Institute for Neuroscience. I have a training in vision science and computational neuroimaging, and I recently started working on methods for what we call neuroinformatics or big data and neuroscience. Tonight, I will show you, I will start very far. I will talk a little bit about the history of neuroscience and then get to the most advanced technologies of neuroscience, and then survey a couple of applications that are close to my heart, and they're part of my close network of collaborators here at UT. So let me share my screen. Can you see the slides? Good. All right, so the open brain. This is the title we decided tonight. And so let's talk a little bit about humans and how complex we are. Both brain and behavior, human brains and the human behavior, are organized at multiple time scales. What do we mean by that? We mean that our brains are fit to cope with events that develop over many ranges of time, from the milliseconds to years, right? So here are the few examples of what I mean by that. In the center is one of us driving through the busy Austin traffic lights. And on the other side, there's a children learning how to read. And on the other side is that process that is happening as we speak to each one of us, which is aging. So reacting to driving and to traffic, it's something that has to happen within milliseconds or seconds, otherwise bad events might happen. Learning how to read or going through school, college, or PhD education takes years or months, in some cases weeks, depending on the case. And processes such as our aging is uniquely for the human life lasts really years, decades. So the brain is fit with mechanisms that actually can support all these very different ranges of behaviors and life experiences. And some mechanisms, such as neuronal synapses and spiking, are best suited to respond to fast happening events now. Other mechanisms, for example, synapses or cellular reorganization, or for example, also white matter and connections, which I will speak a little bit about it later, are perhaps helpful in adapting the brain to long-lasting changes in behavior, in behavioral repertoire, such as reading, learning how to read, or development. So for our luck, neuroscientists are good, and they have developed many ways of looking at the brain. This is a picture from 2014 that was a remake of an earlier picture from 1998. Here on the y-axis, you see different scales of space. This is an axis in millimeters. You see synapses here, the scale of synapses, dendrites, neurons, layers. These are all parts of the basic fundamental building blocks of the brain. And then it goes up all to the brain size, the full brain size. Okay, that's a scale in millimeter. On the x-axis here, you see time, the time scale. You see milliseconds, seconds, minutes, up to hours, days, or months, right? So here, the behavior can be represented, millisecond to second could be driving, and then months and days could be reading, right? Learning how to read. So you see there's each color box, it's a different measurement, and you can see how this measurement very tile the complete picture of the human lifespan, or all the activities the human can do. We go from super high resolution to super slow resolution and low resolution, so that we can cover the whole time. But some of these measurements are perfect for measuring reaction times and measuring events that last very long. And those measurements are best suited here in the green box, best suited best suited to work with animals models. Others, the ones that are non-invasive and they cover often lower resolutions and also both spatially and temporally, are best suited to apply for humans, and they can capture events that uniquely happens in the human life that last, years and months and weeks, like these ones. So on one side, we have measurements that are high resolution that we can apply to animal models that are invasive in that way, and that we can learn a lot from animal models. And on the other side, we have measurements that are lower resolution, but can be, because they're not invasive, can be applied to humans. And because of that, we can learn much more directly from the human brain. So many of these measurements have taken us from this point of view from the brain. This is obviously a post-mortem donated brain to a brain bank that, like a careful anatomist, has detached out of the skull and the brain and the head. And on one side is an intact surface of the brain. On the other side is a picture that has been sculpted with a technique that is called Klinger method, that allows to remove layers of tissue to expose brain connection. You can see here in white long threads and threads of connections that connect different parts of the brain. So these are brain pictures that are difficult to achieve. There's a paucity of brain donations in general. It's hard to get donations, but they're very helpful to understand the rough anatomy. But they're not about you, me, or anyone in the room tonight. It's about, you know, one single individual or a few of them and understanding their anatomy. Well, the measurements the neuroscientists have developed over the last decades has allowed us now to map those same type of properties and of the brain tissue, structure, and function in living human brains. So here I play a little bit around with the words, and we went from brain pictures, the previous pictures, to brain bytes, which is, bytes refers to data. So we have now we've been using the technology that I showed you before to bring this brain from, and our understanding of the brain from pictures and drawing to, bytes and data that can be used and can be reused multiple times to really understand how we work. Now, the marvelous things about brain bytes and digitalization of the brain, which is the process I'm talking about here, is that digital data can go to scale. Indeed, one of the main events in the last year has been the bringing many of these digital images to scale. Now, before we go there, I would like to talk about how science was born, how neuroscience was born and science was born. Bringing data to scale, donating brains to banks, as I just discussed before, it's a democratic process that allows many scientists and students to learn from these tissues. It wasn't always that way. Here's a picture of Leonardo da Vinci. Leonardo is from where I'm from, Italy. And many of you might know Leonardo because of his sculptures or his painting like the Mona Lisa, except that Leonardo was also an engineer and a scientist. Well, it turns out early scientists like Leonardo were not supported by public funding like the NIH or the NSF we have here in the US. Instead, they were supported by wealthy individuals. So in the case of Leonardo, his work was mostly funded by the family that was ruling in Milan, in Italy, and that was called the Sforza family. This is Ludovico Sforza, one of Leonardo's major supporter through life. Now, some of you might know that Leonardo had this very peculiar way of writing. He would write, instead of left to right, he would write right to left, reverse, mirror reversed. Well, until late age in my life, where I am now, I never knew why Leonardo wrote that way. Well, it turns out Leonardo wrote that way because he had no incentive to sharing his data or his findings at the time, because his way of living relied on him being able to deliver when needed to people that would support him and keep his knowledge, scientific knowledge, away from other people that might steal and make a profit out of it instead of him making a profit and a living out of it. Leonardo, actually, it's relevant to what we're talking tonight because Leonardo made a very fundamental contribution to neuroscience. So before Leonardo developed a very interesting method that I'll tell you now, we had this idea that the soul, the intellect of humans was inside the cavities of the brain. This was the major theory of Galeno, another philosopher before Leonardo. Galeno thought that the center of the head has these three chambers that you can see in the picture here, C. These three chambers here contain the soul, contain the connection between our spirits and our bodily essence. And the whole idea was these chambers are in the center of the head. The head is where we understand there is the smart part of us, and then that must be where the spirits, the soul resides. Well, Leonardo actually started smuggling bodies. He wasn't supposed to study human bodies because they were sacred to the church at the time. And so he started smuggling bodies and invented this very clever method to depict the full shape of these chambers inside the brain. These are called the ventricles, by the way. And he decided to pour hot wax inside the brain of a deceased man. And he realized that the chambers actually did not have the shape that Galeno hypothesized. Instead, they had a shape that was quite complex, and there were not three chambers in the center of the brain. Instead, there were these two lateral chambers and two small central chambers connected by an aqueduct, etc., etc., etc. Well, the simple visualization of that new data through that new technique that Leonardo developed as an engineer that he was actually slowly changed Galileo's theory. Galileo was not alive at the time, but the followers studying Leonardo's work and other work that followed Leonardo realized that possibly it wasn't about the cavity in the brain that we should pay attention. Maybe we should pay attention to the tissue around the cavities. So that was for me the beginning of neuroscience, when a simple technological advance made people realize that instead of paying attention to cavities, we should pay attention to the tissue. By the way, that tissue was thought to be just like a motorcycle helmet for the soul before Leonardo did what he did. Here is what I just said. Now, this was not an isolated problem. This is actually an issue that many other scientists besides neuroscience had encountered. Scientists were not initially motivated to share widely their data, their findings, and their results. This is because funding science gave prestige to patrons that would pay for artists and writers and architects and scientists. And because of that, they were not, the scientists, they didn't have motivations to share. Now, things have changed since, and now the modern scientists work for the public good. Modern scientists get funded by the government. They get funded so that they produce things like vaccines that are useful for reducing pandemic and death and in entire countries. And this happened over the last years, 50 years or more, through a process of democratization of education and expansion of education. This is a picture with data from the NSF, and this is the percentage of US population with a college degree from the 1950s all the way to the modern days, the 2020s. You can see that the percentage went from 5% to 35% or more on average. So males and females, it's in different colors. Interesting, if you really look at that graph carefully, you realize that women in 2020, 2021 accounted for 59.5% of the US college enrollment. So there's a major shift in society. There's both a process of democratization of education through institutions like UT, public institutions, but also there's a cultural shift in our society where now males are going to college less than females on average. Very interesting changes happening. At the same time, we can think about higher education like PhD graduates. This is again data from the NSF. And on the x-axis is years, 1950s to 2020, and you can see the number of PhD graduates goes from less than 10,000 in the late 50s to over 55,000 in 2020. So large increase in education. Now, because of this process of education and democratization through institutions and college degrees and PhD programs, many more scientists, or better, many more people have the opportunity to contribute to science and to improve society through science. Well, as part of that process, the community started organizing themselves and developed principles of how scientists should share their assets. Some of the most famous principles are the FAIR principles for open data. How do you share data? Data can mean results, data can mean measurements, data can mean multiple things. How do you share data as a scientist in a way that is fair and democratic, so that other people can have access to it, especially people that are supported through taxpayer dollars to do more good? The FAIR principles say that data that is collected should be findable, the data should be accessible to many people, the data should be interoperable, meaning it should be easy to reuse, easy to take from one system, say software system or hardware system, to other software or hardware system. And data should be reusable, meaning should be very easy to use from a graduate student all the way to faculty in different disciplines or within the same discipline. Here you have the slides, and you can read more about this as you go on and come back to this section of the talk. Now, because of this increasing number of scientists, increase on number of standardization and clarification of the principle by which science should work, neuroscience should work, neuroscience moved to the era of team science or big data or data science. This is an iconic picture from one of the largest projects in the world that is run here in the US by Paul Thompson and colleagues. This is a picture of all the contributors across the world to a project called Enigma. This is a consortium of collaborative individuals to try to tackle important problems of the relation between genetic diseases and brain diseases, but also understanding different psychiatric disorders, such as you see there, autism, addiction, ADHD, schizophrenia, bipolar disorder. Enigma is not the only one. Over the last few years, many large-scale projects for data sharing were actually launched by the NSF and the NIH. This is one of the most famous, one of the beginning of these data sharing projects that the NIH started. This is the Human Connectome project. It's a picture from their website. The Human Connectome, it's a very unique project that comprised of 1,200 subjects in total that were normal subjects. This is a normative data set meant to provide data that can be a solid base for building reference data sets for future studies of disease. And the data set, this was actually a study with a lot of impact, with many publications and extremely important finding that came out of this initial investment. Another one that is more recent is the ABCD study. This ABCD study now will collect over the next years over 10,000 adolescents growing through adolescent, and they will scan using brain imaging technology this adolescent multiple times through their adolescent years. And they will also collect other variables related to their lifestyle, to what they like, how they feel, etc. And all this data will be made available in an ethical way so that scientists will have access to it for proper research. Another very important study that just started is the Healthy Brain Network. It's a mental health study that will help New York children and around the world families and communities to understand how human brains develop and how and when the development is successful and healthy development where sometimes it's not. Now, all these data sharing projects have actually had an enormous impact already, even though I have to say, if you look at them in the long history of neuroscience, it's very recently that we started doing this large-scale project. I would say less than 10 years. This is a nice article that shows the number of publications that use data shared by, this is Michael Milham in New York and other collaborators, show how much all their data sharing efforts affected scientists, even helped scientists to publish in peer-reviewed journals, solid publications. Similarly, Alzheimer and dementia are key aspect of society that we're trying to tackle as scientists, and similarly, this type of initiative had enormous impacts on clinical trials and on improving understanding of Alzheimer. And this is another report that was made for showing the impact of that project of data sharing for Alzheimer and dementia. Here's the number of publications reusing the data, and over time since 2008 when the study started, and this is a publication from 2014, actually 2015. Now, data sharing has become a large aspect of what we do. I also partially work in collaboration with others to make sure that data is fairly shared. Now you understand when I talk about fair sharing of data. And these are some of the major data archive that the NIH and SF and also other similar brain initiatives in Japan, in Europe, have made, supported, have funded, have put money on the bench so that researchers could put data on these archives and then they can reuse the data. By putting data on the archive, what I mean is that there's files that we can collect by doing different, using different measurements modality, just like the one I showed you early on in the picture. And these files live on hard drive, just like the computers we're using now here to listen to this talk or to make the talk. And these files are specific to different data modalities, and scientists are very democratic and put them onto archive where other scientists can go and use them, reduce them, so that we together understand more and understand faster about the brain. Okay. Now, you're probably thinking, wow, there's a lot of archive. You already talked about many different types of projects, and you showed us a picture of many different individuals and centers in the world. And like, this sounds very complicated, and indeed it is. It is very complicated. It's a problem that of how to share data fairly, how to share data ethically. It's a problem that many of us are tackling. And one of the problems that comes attached to the sharing of data is the fact that data is not enough many times. Indeed, for scientists to use data, the data needs to be changed, denoised, massaged, ameliorated, made better by software. And software runs on hardware. So in fact, here it's another article, a picture from an article from my two collaborators, Yaroslav Halchenko and Michael Hanke, that make the point that we need not just data, but we need data and metadata. So data and software records associated to the data, so that we actually know how to use the data for benefit of society. And data and software actually need computers. And this is a picture from one of the top supercomputers in the US, Frontera, that is here at TAC Supercomputing Center at UT Austin. And Frontera allows many of us to process thousands of brains in a heartbeat. And this is, again, one of the good things that support to science does for society. Supercomputer like Frontera exists because the NSF funds them, and for research and for researchers like me and others. So we need a good, willing culture for sharing. We need data to be collected, and that's costly. We need data to be shared. For that, we need systems to host the data and process the data, and we need software. Many things have to come together for all this era of big data neuroscience to be a realization, be real. So here I present another project that is funded by the Brain Initiative and NIH, is run by my collaborator, Russ Poldrack at Stanford University. It's called Open Neuro. Open Neuro collects, it's a data archive, is one of these examples, a key example of data archive. It collects data from different data neuroimaging modalities, MRI, magnetic resonance imaging, PET, positron emission tomography, MEG, EEG, and intracranial recordings of electrical activity in humans and animals. And you can see here one of the interface. The archive looks like this now in the latest version. You can go inside and you can see, each one of these subject 1, subject 2, subject 3. These are all files that some good-willing researcher collected. In this case, this researcher, I actually don't know them, but I believe they are in China, and they share data on loss enhancement and then layer selective signal in specific pathway of the human amygdala, which is one of the key, amygdala is one of the key components of the brain for regulation of emotion. So they share the data, and I can go as a researcher and download the data or move the data to a system like Frontera and do processing of the data. And this is difficult data to analyze. Now, this is another example of modern technology for large-scale data analysis and insight generation. It's called Neurosynth. Is currently run here at UT Austin by Alejandro de la Vega, one of the panelists tonight. You get the chance to ask questions to Alejandro later. Neurosynth is slightly different. Neurosynth collects information from papers. Here is a picture of the workflow of Neurosynth. Neurosynth synthesize knowledge from papers, from published papers, and projects that knowledge onto brain structure. So for example, you can ask questions, pain, and it will generate from you by searching through papers, right? It will identify the locations of the brain that are relevant to the term pain from studies that are published and studies that contain coordinates of locations of activation during pain stimulation in imaging studies in humans. So it's different. It's not an archive of data. It's actually a metadata system. It's a system that analyzes metadata and generates images of data of the brain from published literature. Very interesting, clever. These are other pictures of how, if I do a search for emotional, here search for emotional, turns out that the system Neurosynth finds location of very common activations due to emotion. And as it turns out, this nice red dot, the activation, lay perfectly on top of the amygdala, the location of the amygdala in the human brain. And that, as I said before, it's a core system for emotional regulation. Another example I will give you today, I'll spend a little bit more time on this one. It's technology that we have developed in my lab, which is called Brainlifeio. What I showed you today so far is that there's projects dedicated to data sharing. There's projects dedicated to data archiving. They act as archives for data. And then there's a need for computing, and there's a need for software for analyzing the data. So in my lab here at UT now, we've been developing this project funded initially by the NSF, and now we also receive funding from the NIH to integrate. This is called Brainlifeio. It's a platform, it's a website that you can go, just like you go onto Facebook, right? This is Facebook for scientists, right? Scientists can go there, instead of sharing pictures or sharing analysis, they can share, instead of sharing pictures or sharing text, they can share brain pictures, and they can share code to do analysis, right? And the platform connects with the supercomputers and connects with Frontera, connects for Jetstream, which is another major project funded by the NSF and supported at Indiana University and other partners, PAC Supercomputing, one of them. And so the platform brings together all the different aspects of all the work I've been showing, the code for analysis, the data, and the supercomputers. So it's a public and free cloud platform that supports reproducible neuroimaging research. It integrates data management, processing, and visualization, connects code wizards, people that are good with code, with data gurus, people that are good at understanding what neuroscience need to address societal needs, such as understanding disease, with supercomputers. It promotes data sharing, code sharing, and tries to support advanced, difficult scientific discoveries. There's multiple data modalities that we currently support. All of them come from magnetic resonance imaging, anatomical MRI, functional MRI, and diffusion MRI. Anatomical MRI is about brain structure and anatomy. Functional MRI is about activity as you do tasks or as you rest in the scanner. Diffusion MRI, I'll talk more about that later. It's about brain connection, understanding brain connection, connectivity, and the brain really needs proper connections to work fine. So many types of data are generated through the platform, and these data sets have been benchmarked over the years now, the last 3 years, to try to generate important scientific results and insights. And many people have collaborated in the project, and we're pretty happy with where we are at this point. So I'll give you a quick glimpse of how the platform looks from the inside. Here, I just logged into the platform. I'm looking at different projects. There's many projects that people are working on. This is one specific project that one of my students, Bradley Caron at Indiana University, he'll join UT in January, has been working on. He's been analyzing many data sets. These are all visual interfaces, right? And I mean, they're not quite like Facebook. I'm sorry if I said that before, but they're exciting for scientists. You know, they allow visualization of data. Look at this, right? So here we went from files on a system, and without downloading or running the file through a different software, we're now visualizing brain connections from a living human brain that was collected. Who knows? Maybe this data was collected at Stanford, or was collected at Harvard, or collected at a different institution here in the US or outside the US. In fact, many people seems to be excited about the platform, and we have seen over 2,000 logins at this point, and we have about 400 active users, and hundreds of them are active every week, and they're using the platform for brain analysis and understanding the brain. This is a little bit older slide here. Now, here it's another picture of how the platform is different from an archive, in the sense that we connect with archive. Here is an interface that connects us to Open Neuro, what I showed you before. I can go through or you can go through this interface, and you can grab data from Open Neuro, import it into Brainlife, and then create a private project or a public project. Most of the users create private project for many purposes of ethical purposes and reasons. It is more important. It's better to do that way. And then you can analyze the data. You can visualize the data. We serve a variety of visualization software that other researchers in different parts of the US or outside of the US develop. So data from archive become actionable on cloud platform like Brainlife. So we go from static file that you've seen before to living file that can be used for research. You can visualize them, you can analyze them, and you can try to get insights out of the data. Here is another video in which I go through how the data processing can be done. These are a few examples of applications for data processing of the data. So here, it's a functional MRI preparation, pre-processing application. This is developed by Oscar Esteban, one of my collaborators. And in this case, we took the code that was developed as a library of software by Oscar and collaborators, and we made it into a living service that actually exists and can be used by users. See how many users at this point were actually using that app. And here you see, you can select data sets from projects. Here I'm selecting one project, and then I try to submit the data set, and I can analyze the data by choosing the software that someone, say, in Switzerland developed, and taking the software that someone in the US developed and putting them together in the cloud, and then the software and the data get deployed, say, on Frontera or on Jetstream, and you don't have to know how to do all that. Right? You can do these analyses and you can do this processing properly using the proper software, the proper data on the proper system, just focusing on what is that you care in terms of science and scientific results. So the software comes alive as cloud services. Cloud services is modern technology that we have nowadays. That is the same technology that is behind Twitter, the same technology that is behind Facebook or Google. It's a service means that you have a point of access to software through a web browser. That web browser serves you software that runs, so that does operation for you. For example, you can share your pictures on Google Photo, or you can share, or you can grab your documents from Microsoft documents, or etc. Or you can share pictures also on Facebook or tweet on Twitter, if that's what you like to do. Now, so far, I went from the history of neuroscience, the basic measurements of neuroscience, down to technology. I started from Leonardo's technology, which was this clever method of putting wax inside brains to give a picture, complete picture, to the most modern technology we have, which is the cloud technology, right? Technology that runs in the cloud, that runs on supercomputers without us having to actually get close to the supercomputers. But how can we use the combination of this data, these many measurements that neuroscientists can do, and this cloud technology to make an impact to understanding clinical disease, and then try to understand basic principles of how the brain works? Well, to get to that, let me step back a little bit from the measurement. Let me talk a little bit about the brain. So the human brain is organized just like the internet. Okay, we often talk about gray matter and white matter. And gray matter is here where the purple is darker, more intense purple. By the way, this is a very good old stain called the Nissl stain. It's a stain that's stained for the bodies of the neurons in the brain. So where there's a lot of bodies of neurons, you will see dark purple because that's where the stain goes. And when you see lighter surfaces, that's where the brain connections are, like long connection between neurons. Don't have many cell bodies in there. There's just axons of connections. So here is for the cables, and here is just an example of a cable. Why am I calling these computers and cables? Well, you can think about the brain just like the internet. You can think about the brain as being composed by computers, such as the one on your desk now, and then cables that connect to the internet through other computers, just like the one on my desk now. Right? And these cables and the computers have to work together for the brain to work. Think about it. I can have a very fast computer on my desk, but if my cable breaks or if my cable gets really slow, I am not going to be able to deliver a talk tonight. Most of what we do nowadays requires good computers and good cable connecting the computers, because we are living in an interconnected world. Well, it turns out the brain is just like us. The brain is an interconnected world of computers, the neurons, connected through cables, the axons. So sometimes cables are like copper, and sometimes cables are fiber optic. And you might know that fiber optic is better at conducing information, less loss of information, less reliable to humidity. Copper, it's good for short connection, but you can't use copper for very long connection unless you start thinking really hard about the engineering of the cables, right? So, and the same is for the brain. Some individuals, for some connections, have something that looks like copper, or maybe during development, something that should become as good as fiber optic actually ends up being copper. And then the child has a problem, and we would like to understand, can we do anything for this child so that the copper cables turn into fiber optic cable? Because whereas cables are just made out of static material, the human brain has a lot of plasticity to it, especially early on in life. So brain pictures is what we started from, and brain bytes is what we're talking about, right? This is what we can measure with modern technologies. But how do we get to these brain bytes? So let me give you one example of one technology that we use from brain bytes. So here is a brain picture. It's I said before, this is a brain that was sculpted with this Klinger method and allow us to actually see, you can see here, one of the major connections. I call this a highway that goes from the visual cortex in the back of the brain deep into the temporal lobe. You can see it's hard to see, but I'm used to this, and you can see cables go in that direction. There's another one that it's possibly the cingulum that connects deep into the temporal lobe, into the frontal lobe, and also the anterior thalamic radiation that connects the thalamus to the frontal cortex, and then the corticospinal tract. The corticospinal tract connects our motor cortex down inside the spinal cord and allow us to move and control movements. We can see all this, but if you're lucky and you have a scanner, this is a Siemens scanner, one of the most used nowadays scanners. And this scanner allow us to take pictures of living brain that look just like this one. Obviously, this picture is not from a living brain, but this one is. This one is a picture of the same tracks I just showed you before, connected in a living brain. This was my brain when I was a postdoc a few years ago, when I was still younger. And this picture of the human brain, it can be used because I can take a picture of myself again today, tomorrow, here at UT. And I can compare that picture that is now 10 years old with the picture today, and I can look at my process of aging. I can see how my brain has slowly changed, and probably that change will continue. But that is good. We can measure with this technology tissue properties, connections, cables connecting computers. We can also measure neurons, the computers, and try to understand how and when and why something might go wrong during development, during aging, across the lifespan. Now, how do we do that? So, well, it turns out these connections, just like the one that I showed you before, actually are made out of little fascicles, bundles, just like little bundles of salad. Here, it's a picture of the optic nerve. Here, from top to bottom, you see little connections that were cut of the human optic nerve, okay? And you can think about that these connections are made out of material that are biological tissue, but also they're filled with water. And water in these connections actually moves freely in the direction of the connection, but it doesn't move as much freely against the direction of connection. Well, it turns out MRI has a technology that allows us to measure water diffusion in different locations, and then we can identify from that measurement of water diffusion whether the connection were going the blue direction or the orange direction, up or down or left or right. And by measuring in multiple locations in living human brains, we can actually measure the directionality of these connections, and we can reconstruct them long range. So what we do, we just a brief idea of what we do. We collect pictures like these ones. This is again from my brain. We fit models to them that represent the connection directions at each location of measurement. And then we use computational methods, we use computers to track, to connect these dots, right? These biological dots are connected so that we can reconstruct pictures of trajectories of major connections in the human brain. So these are a few pictures of my brain, again, my major connection. This is a connection that goes between the back of the brain to the front of my brain. It's called the inferior frontooccipital fasciculus. Very important in connecting vision to control and inhibition. This is another picture from my cortical spinal tract, left and right cortical spinal tract, reconstructed with this method of MRI. Once you get these pictures, this data, remember, these are 3D rendering of data, of biological data, we can actually look at the property of the tissue, so the cables, remember, and we can see whether the cable looks more like fiber optic or more like copper. Is it good tissue or is less good tissue? And we can try to think about why is Franco's cortical spinal tract doesn't look as good now as it looked when he was 20 years old. So in a certain way, we're going from preparation like this one. That's where Leonardo started. He was one of the first to be brave and knock at the door and smuggle bodies to understand neuroscience. He was one of the first. Started from tissue preparation post-mortem, from like these ones, to beautiful picture created with super fancy technology and supercomputers like this one. This is just a series of brain structure with the connection passing through them that we created using Brainlife. Now, I'll give you an example of what we can do with this sort of technology. This is a paper that we published now a few years ago that reviews how vision is served by brain connections. Human vision is really important. And it turns out the two arrows there highlight the major connection between the eye and the cortex, the visual cortex. Visual cortex is in the back of the brain, which is down here and down here. And here you see the nerve. You've seen a picture of the optic nerve before. You can imagine the eye here are missing in this resection. This is the trajectory of the nerve into a first stage in the thalamus, and from the thalamus, there's connections that go into visual cortex down here. Well, using technology like the one before, we can measure that structure. We can try to understand how that brain structure is affected by, for example, eye disease. In this case, we study with these collaborators of mine in Japan, age-related macular degeneration. That means it's a disease that affects the macula, which is the center of the eye, where we have the highest resolution for vision. And I'm going to show you now some data from the eye first. So that's where we're starting. So that is the fundus of the eye. Here's a picture of the fundus of the eye. And here where the arrow is, it's where this individual, it's one eye, one individual. This is, for example, the beautiful vessels getting into the eye. And you can see here is where the optic disc is, where all the nerve, all the cables out of the neurons in the eye. Yes, we have neurons in the eye. Okay, your eye is a very sensitive instrument. It's full of neurons, just like the brain. Okay, the neurons die in these locations here. And because the neurons die, we lost vision. And here, it's a picture of how there's a loss, there's a thinning in the same location. There's a thinning of the eye tissue. And because of that, this individual doesn't see very well. So this is a representation of where the individual sees better. Black means this individual did not see very well. Gray saw a little bit better, but still not very good. And then here, some residual vision was found. Now, macular degeneration affects the retina. The question is, does it also change your brain as you lose vision from the eye? How does that affect your brain tissue? Well, I showed you before that this is the pathway that the information from the eye takes to go to visual cortex. And we were able, using the technology I showed you before, to reconstruct those pathways, right? But also, more importantly, we were able to separate the connection that go from the center of the retina, here in blue, from the connection that goes to the periphery of the retina, here in purple. This was unique for this study. We were able to separate which connections went from the macula in the center of the eye to cortex, and which connection went from the periphery of the eye to cortex. And we tried to track those connections to show whether the effect the loss of vision in the eye affected visual cortex and the connection in a very specific way, right? So if there's a trauma in the center of the eye, we were expecting a trauma in the corresponding location in cortex and in the connections. So here is a plot of what I just initiated for you. Here is this part of the connection. This part is called the LGN, here the LGN. And here is V1, here is V1, visual cortex number 1. So here I'm plotting the property of this connection from the LGN in the front down to V1, from the LGN down to V1. And I'm plotting here a property we call fractional anisotropy. It's a summary how much water is going in a single direction versus all the possible direction of water diffusion, if you remember when I talked before about water. But, you know, what is important about that connection, it's a proxy for the quality of those cables. Is the cable more like copper? Fractional anisotropy is down to 0. Is the cable more like fiber optic? Fractional anisotropy is high. So what you see here in black, you see the controls, right? The controls in black. You see that the controls have higher fractional anisotropy, meaning they're closer to fiber optic than the patients with macular degeneration. Now, this one are the fibers that project to the macula or fovea, the center of the eye. But if we look at the fibers that connect to the periphery of the eye, now you see that difference between the colored line and the black line disappeared. That means that the damage to the eye due to the disease did not spill over to fibers away from the fovea, only affected the fovea. And here is some mid-region where the fibers connect closer to the fovea, and still we find no difference between the periphery and a major difference in the fovea. Also, it turns out that that property, fractional anisotropy, connects with the visual acuity, how fine of the detail a person can see. So individuals that have higher acuity see better, so they have more spare vision in the eye. They also have better properties of fractional anisotropy in the same corresponding connections. Here's another picture of that. We've done several of these studies with many of my collaborators, both when I was at Stanford, still working with Brian Wandel, and also with other collaborators that over the years here. This is one of the recent done with one of my postdocs at UT, Sandra Hanekamp. What we find in general is that when there is an effect of the eye, your brain changes. This is not something we think about, but as you start wearing glasses or start having eye problems, your brain doesn't stay the same. It's actually affected by the reduction of inputs from the eye inside your brain tissue. Cables change, computers change, many things change in your brain as a result of your loss of vision. I'll show you quickly another study from another collaborator. I talked to you before about development, and this is a study about development that Sophia Vinci-Bucher, postdoc in the lab, has just completed. Sophia is interested in how different parts, different connections in the brain are affected during development, how they develop over time. And there's a major theory that we understand the brain has a pathway in the dorsal stream, we call it here in gold. That pathway supports vision and action, whereas a pathway in the ventral stream here, in this part of cortex, it supports vision and object identification or generally perception. The question is, okay, what are the connections supporting the two pathways? How do they develop? And importantly, how do the connecting between these two pathways, here in green, how do those connections develop? Sophia found out that the connections that connect up and down the dorsal stream and the ventral stream actually develop after the connections for object identification, but before the connection for vision and action. Okay, that's her main result. And here is how she used the technology of Brainlife I showed you before to actually identify those connections and then plot the properties of those connections and compare them between adults in black and children in pink, etc. We've done similar study. We collaborate in different species. These are now brain from, here is a human brain, here is macaque brains, and here is also a marmoset brain. So we've done some experience working with different image modalities, because this is the beauty of all this technology that is coming to us, allow us to compare from animal models to human brains and bridge that gap between species so that better understanding of a better picture, clearer picture of how the brain in general works. Beth Lippard is assistant professor and one of my collaborators here at UT. She was kind to share some of her slides. Beth addresses really important societal problems. She's trying to identify risk factors in bipolar disorder. She's trying to understand neural, behavioral, and familial risks. What are the precedents that we have in our genetics, in our brain, and also in our background as families that might support bipolar disorder? So bipolar disorder is really important because it actually leads to suicide often. Indeed, over 40,000 Americans every year will die from suicide. That is a very sad event for families, disruptive. And the trends seem to be stable over the past 10 years. So this is a strong problem, big problem. And turns out suicide is associated with mood disorder, and bipolar disorder is the highest suicide risk for an individual, especially here you go for adolescent. Indeed, this is an axis that's across age, 1 to 4 years, 5 to 9 years of age, etc., up to over 55, 65 years of age. And this is the number of deaths in the US by suicide. And here what you see is that the attempts, it actually grows exponentially between age 14 and age 34. So this is an event, suicide, that often has grows exponentially during adolescence. And Beth is trying to understand how dynamic brain changes in frontal cortex and in the limbic system, for example, synaptic pruning, changes in connections, changes in myelination, which is what we were measuring in the previous study, and how those changes during adolescence predict through adulthood. How do they predict, risks for suicide and risk for increasing bipolar intensity? So she's studying circuits that connect the amygdala, again, that circuits that connects the temporal lobe and emotional center, to the striatum, that's a center related to evaluation and pleasure and feeling satisfied in life, and the frontal cortex, which is emotional regulations and also impulse control that sometimes goes wrong, especially in adolescents. So these are the connections that she's interested, connection between the amygdala and frontal cortex, striatum and frontal cortex. And these connections might be key in understanding bipolar disorder, and also how emotional regulation might grow better or worse in adolescence, and might be a risk to observe and monitor in predicting potential suicide attempts. So here it's different pictures of how also the volume of the structure, amygdala and frontal cortex, changes across adolescence. So that's also something that we can track with these technologies. And we can see that in bipolar disorder, often the volume decreases during adolescence faster than the normal rate, okay? And the volume of the amygdala stays lower across adolescence. The key connection that Beth is interested in working on is what I mentioned before. It's called the unsynaptic fasciculus. It's known for many years, but we're still trying to understand the role of these unsynaptic fasciculus connecting the amygdala to prefrontal cortex, and how the white matter, so the connections, whether the connections are more copper-like or they're more fiber optic-like, and whether they're different enough between individuals so that we can make predictions of whether this individual will develop bipolar disorder or will be a risk for committing suicide. Very important research, and these are some pictures, some results from her. Indeed, she finds that there's an increase in some of the tissue properties in individuals, normal individuals, and then here the difference between healthy control and bipolar disorder, it's big, it's relevant, significant in precisely these areas of connectivity between the amygdala and prefrontal cortex. Now, this is to wrap up. I showed you now, sorry about that. I showed you now, I hope, an interesting path through the history of neuroscience, how neuroscience went from being a closed system to an open system and an ecosystem that uses technology to actually generate, turn pictures into data through technology, and share data and reuse data and create insights out of data to address important societal societal understanding and help with cure and help with disease. This is a picture that shows some of the data that we can generate with these new technologies. Here are some of the brain connections we have, and I want to thank the team of the UT by Brainstorms, Mike for inviting me, and I'm looking forward to meeting the panelists. I see there's some pictures now, and what shall I do? I want to thank my funding here at the end, but also that's what allows me and my lab to do the work we do. And this is public funding from the NIH and NSF. And we also received some funding from Microsoft.
Okay, Franco, great. Thank you so much. Why don't you introduce your panelists and get everybody on board here as we queue up the questions?
Excellent. So shall I stop sharing my screen?
Please.
Great. So all the panelists, you can now turn on your camera, I would say. Hi, Beth. Hi, Beth. So let me introduce you one by one. So I'll actually start with Beth. Beth is assistant professor in psychiatry at UT Austin. And Beth uses neuroimaging to study brain development and early illness in bipolar disorder, with emphasis in development of suicide thoughts and behaviors to addiction. Beth, you've seen some of her great research data and results. We also have in the room Dylan Kirsch. Dylan is a neuroscience graduate student, so she's a graduate student in the Institute of Neuroscience here at UT. And she works with Beth, and she's interested in using neuroimaging techniques to understand alcohol use and brain development in young adults with psychiatric illness. In the room also Alejandro de la Vega. Alejandro is a research scientist in the Department of Psychology, and he is one of the gearhead behind some of the tools you've seen before. You've seen Neurosynth, and he's leading the lab that is developing that system. That you remember that system that allows us to go and query from text to brain data, brain results, right? So Alejandro is in the room too. You can ask questions about that. And then Hao Ting Wan, I'm very thankful for you to join us. Hao Ting is joining us from Montreal. She's postdoc in the Montreal Geriatric Institute. I'm sorry about my accent. And Institute at the University of Montreal. She works on neuroimaging software and Alzheimer's disease biomarker. She wears two hats. On one hand, she's trying to understand disease and how the brain works. And on the other hand, she likes to develop software, like many of us in the room. Annibal, last but not least, Annibal Heinsfeld, he's computer science PhD here at UT, and Annibal works in my lab currently trying to use AI systems to understand human vision and then make actually some of these platform works better. That's where his focus is. Thank you guys for joining us. And now I guess we can address some of the questions.
Great. So let's start with a question from a longtime Brainstorms participant, Joe Virgil. Joe asks, can you, given that you have data from all over the world, can you see differences in brain data from people from different cultures? So are there sort of cultural markers that one can see in the brain?
So that is a very interesting question. There are, and sometimes they're cultural, I would say ethnical. The amount of data or reports that look at things sensitive like this question are limited. I mean, to my knowledge, there's few, but there hasn't been an enormous amount of data looking at ethnicities and differences between ethnicity. It's probably the case that there are differences between different ethnicities and different culture. Now, how they impact life, how they impact disease, how they impact normal living of individuals. It's not something that we have addressed enough. In this, there's, related to big data, there's now an organization called the International Brain Initiative that is trying to bring together individuals from different countries so that more data from different countries and cultures are shared so that we can actually start tackling questions like this one at the right scale with the right meaningful answers.
So there was a brief question about which of the 3 MRI techniques would you use to detect a stroke?
Actually, FLAIR images are used often for detecting a stroke. Diffusion MRI is also being used for detecting a stroke, where FLAIR is what is commonly used in clinical settings. Diffusion MRI has been used in clinical setting, but primarily it's in constant development through research. And there's a lot of hope for understanding stroke through diffusion MRI.
So Scott asked several questions, and the one that really caught my eye is that he's asking about how brains, you mentioned that brains can react to different time scales and statistics of the world. And Scott asked, do you think that our modern world of constantly switching contexts and such, is that affecting the way our brains operate?
The answer is yes. It's not just an answer from, my point of view. And I hope, maybe Beth and Alejandro and you guys in the room know more about that too. But I've seen now the first reports of how individuals that engage more or less in task switching, that is the technical term in neuroscience, task switching between tasks, switching between phones, computers, and one screen and another screen. It really drives down your cognitive ability, and that's possible. That might change in the long run, your brain tissue and brain function. Beth, do you know anything that maybe I missed?
I would have said exactly the same thing.
So one thing I would like to add, though, is one thing that's interesting is to actually study that question is not that easy. And in part is because a lot of the measures that we use in the lab don't necessarily reflect the real world as much as we'd like them to. So that's actually one thing we're looking at in our lab. We're trying to use naturalistic data, which is trying to be a little bit more like the real world, like complex movies, for example. But that has a whole range of challenges to analyze because the data is so much more complex. So that's another way to bring it back to Franco's overall theme, that this sort of, enabling cloud technologies and, large scale processing might help us wrangle. And a lot of times we're constrained by what we can do, what we can analyze. And those questions in the context of a very realistic setting are not easy to answer, but we're slowly making progress.
That's a very interesting and important point. Let me follow up a little bit on that. So what Alejandro is bringing up is the fact that a lot of science happens in labs, right? And that is important. That is very, very important, right? Because labs have the right conditions for us, experimenter, to create, the preparations we need and the observation we need. But often what happens in lab is far from questions that the population or the citizen like us would like to address, right? So unfortunately, a lot of the science we do answer fundamental questions that are science, but cannot directly address questions that might be relevant to the world. So there is a movement in neuroscience to try to start thinking how we can use modern technologies, right, to develop different ways to think about data so that we can almost hijack the problem that we have and get closer to real life, right, by using, for example, stimuli that are videos and they're natural. This is what Alejandro's lab is doing, and this is something that also Annibal is looking into. How can we use, right, more natural stimuli instead of using conditions that can only be done and replicated inside, a room, a UT, or a Harvard, or a U, etc. cetera. So that's, yeah. Alejandro, do you want to say something about what you guys are trying to do specifically?
Well, we're sort of, luckily I've been working on this all day, so it's fresh in my mind. But there's two challenges. One of them is that the data in the actual movies is extremely complex. So there's so much going on at once to actually isolate a specific process that we can say with some definitive answer, this region is correlated with, for example, speaking of switching, you can identify when the scene switches and there's a camera change. To actually annotate every single time that happens in, for example, Raiders of the Lost Ark, which is actually one of the stimuli that we use in one of the studies that we have, that would take a lot of hours to annotate that. Now you also want to annotate every single other thing that is relevant. So speech, you want to annotate the visual properties, you want to annotate object categories, like there's people on the screen, there's different objects on the screen. And this is simply difficult to do manually. So we're trying to explore ways to use machine learning algorithms to automatically give us features to look at. And what we really want to do is we also, one of the things that Franco said is there's a lot of data sharing going on now. So we're actually applying this on public data sets. And we really want to be able to do it on a large number of data sets to actually give us a more precise answer, to give us a more confident answer. So that really becomes difficult. We're trying to do it across, potentially, dozens or hundreds of hours of movie watching. So this is really where technology for both the data analysis of the imaging data and also the preparation of the stimuli to actually understand what's happening in the stimuli makes this possible.
We're not there, right? We're not there. But imagine a future where, we're all watching a movie on Netflix and we're wearing a little device, and then suddenly, Alejandro's future Alejandro algorithm will detect that some of us have a problem, and maybe there's a phone that rings, and then we get to talk to our doctor before maybe a stroke arrives, right? So that's one way to think about what Alejandro's team and this new technology could do, right? Could monitor us on, online, and by sending data, small snippets of data to the cloud platform can process that data and send, the information to the right healthcare provider so that, we can figure out something's going on or not with you.
There's another question that I could summarize as saying that in other fields of science, people are developing models. And the question is this something that's happening in neuroscience as well? Are people building models of the brain? Is the idea to develop an interactive model of the brain? And before you start to answer, I want to say that just to advertise that if you go to our YouTube channel, we've done a couple of these. One of them was the artificial brain, and we talked about the possibility of having a machine that would have a mind, and it was really just an excuse to talk about computational neuroscience.
Well, I almost wish you answered that question, but I'm going to take the first step. So Mike here in the room is probably the most expert modeler, but yes. So the ultimate goal of neuroscience is to use this data to build predictive model. Okay? And predictive model are models that embed a theory in them. They're models that understand how a system and a process works in the brain so that we can make a prediction. Now, the prediction could be interpretable or non-interpretable. And, an interpretable prediction is something for which you understand the fundamental processing within the model. And then non-interpretable prediction is the one I made an example before in the case of Netflix, where there's some AI system that kind of munch some data and then detect something different and send it to the cloud, right? So yes, there's both ongoing processes. There's people like Mike, and and sometimes I wear a hat in which I develop also models and with some of my students. But there's people dedicated to, there's a whole field called computational neuroscience that focus on developing models. Mike, do you want to say something about that? That's pretty much your field.
Well, we've done past brainstorms. We're going to do future one on there. So I would say stay tuned. We have some coming in the spring maybe that will be about that. So.
But in general, right, every field of science, just like neuroscience, is about building an understanding that we can embed into a model, that we can specify to the degree to which, so that we can build a little piece of software, a device, and that software and device can be, generally applicable to different cases, right? So generalizability through modeling it and understanding is key also to neuroscience.
So unfortunately, we've hit the time of night where our time is running low, and we've done this thing now for a while where we have the panelists, we give each of the panelists a minute or so to tell us something that they haven't had a chance to say. And with 5 panelists, we'd better start a little early. So let's just go one by one. And so the panelists, the challenge here is that you have a minute or two, maybe closer to a minute, to tell us something that you haven't had a chance to say because there's just too much to talk about tonight. So Hao-Ting, would you like to go first?
Yep, I will have a head start. So the questions from the audience are very interesting. And the part of work I do combine a lot of data from different research centers. And that has helped us to understand our brain in a wider context. When we are collecting smaller data set, you have people about similar age range, but now we have access to people with a wide age range of age. And now we can, in some way, understand how our brain is functioning in a more realistic context of like how it represents the population. So that's a different impact that opening up these data is bringing to us.
Great, thank you. Annabelle, would you like to go?
Of course, yes. So following what Hao-Ting was saying, I guess much of the clinical applications for such technologies, they are very important, and this data sharing, it's very important towards that, because then we have this variety of brains, because the brain of people, if you look from the outside, it looks the same, but when you start to see it functioning, it can get really, really different in general. And having this sort of data sharing and collecting all this data, it's definitely a good input as a source of variability across the whole world population, let's say. And another thing towards maybe I'm crossing my time, but towards the simulation, one thing that neuroscience suffer is to measure the brain functioning. And because our methods are still evolving, and we still cannot measure at the level of all the neurons in the brain. So a dream that I definitely have for cloud computing is to be able to simulate a whole brain in large scale. That would be amazing, but that's still a dream, a long, far dream.
Great. Well, we can all dream about that. So, Dylan, what would you like to say?
So I guess kind of building on what Annabelle just said, a goal in clinical neuroscience is to use these neuroimaging techniques to detect early markers of illness, to guide early intervention before disease progression. And oftentimes the earliest markers and mechanisms of disease are very subtle, especially early in the course of illness. And so big data and open data can allow for larger sample sizes that are necessary really to identify these more subtle markers. And also kind of something to just end on that open data makes me think about is this concept of wisdom of the crowds, or the idea that large groups of people collectively are smarter than an individual expert. And so when it comes to problem solving and innovating and predicting, and so I think that open and big data can really allow for more creative and innovative science that allows from scientists from different backgrounds and expertise to work together.
Dylan, can you give us an example of what you mean by a marker? Because I think that's really cool.
Yeah, so subtle differences in brain development of certain regions that are implicated in psychiatric illness, for example, bipolar disorder. There's been increased study of early detection of these diseases early in adolescence so that they can be targeted and individuals can be treated prior to illness progression. Because as shown in some of Dr. Lippard's slides that Dr. Pastelli presented, many of these psychiatric illnesses are really progressive. And so early in the illness course, differences in the brain are really subtle and much harder to detect. But it's been shown that if you can detect this and treat this really early, people can do really well, and you can ameliorate some of the more progressive decreases in brain differences in illness.
All right, well, hurry up with that, please. Alejandro, what would you like to tell us?
Just to add to bring it back to Galileo, Leonardo, I think the one highlight for me is to remember that having open science is a goal that we're always expanding on. And it's, maybe one thing that the public is not aware of is that sometimes it's in conflict with some of the goals we have as individual scientists. So luckily, I think there's a lot of people here and generally in science working on tools to make it easier to share your data, make it easier to work openly. And in the end, those ultimately are going to help reach some of the goals, for example, that Dylan was saying, that actually have an impact on people's lives. And so that's really the goal of a lot of the work that we're doing.
Cool, great. So Beth, in baseball, these days, they always put your best hitter fifth in the order. So why don't you go fifth and close this out with something really cool?
Oh, we're in trouble. But I guess if I'm gonna, end it out, I would piggyback on what Annabelle and Dylan was talking about, the clinical relevance. And I think Dylan did a really nice job talking about how there may be subtle changes that could be a biomarker that we could identify risk to intervene early. But adding on to the complexity, oftentimes, right, the risk factors in one individual is not the same for all individuals. And so if you just take it to the context of suicide is a major problem. It is preventable, but we can't predict it. And so there's a lot of groups. Franco actually mentioned the Enigma consortium, and there's one on suicide that we contribute data to, because suicide are really rare events. And so if you want to think about individual differences, if you want to think about risk factors, we need really large data sets so that we can subtype risk, because the risk factors, they're not going to look the same for every individual. And as a scientist on my own, I can do a research study and I can have this small group of people that I have money to actually scan and look at behavioral and clinical data on, but it can only get us so far. And so really, I think the innovation that's going to come over the next couple of decades is really going to be through open science and from scientists collaborating with one another to answer some of these really complicated and important questions.
Great. So Beth, thank you. Alejandro, thank you. Dylan, thank you. Annabelle, Ting, thank you so much. And of course, Franco, most of all, thank you so much for a great Brainstorms. Fantastic. Just close by thanking the people who have been here tonight. And just as a reminder that we generally skip December in Brainstorm. So our next one will be January 27th, and we'll be posting the schedules for those shortly. So thank you, panelists. Thank you, Franco. And great night. And we'll see you all soon.
And happy Thanksgiving to everyone.
Thank you so much, guys.
Yes, bye.